Hi, everybody. We've got a full room here in Boston. We've got about 100 folks on the Zoom. As a reminder, we've got 4 companies presenting. You guys go around the side. We've got 4 companies presenting here, and each one will have 10 minutes to go through a handful of questions that we've given them. And the idea is to whiteboard out the brass tacks of your company. And then there'll be the opportunity for people to ask questions. If you have questions, you can put them in the Slack. If you can't find the Slack, just pop in the Zoom chat and we'll show you where to go. And we will throw some of those questions out at the end as part of a panel. Sound good to everybody? Great. So they don't all respond at once because it'll get very loud. All right. So we've got Manifold getting us started here. Shane has bravely volunteered to go first. We really, again, appreciate everybody coming. And as you have questions, submit them via Slack. If you're here in Boston, just shout them out when we get to that point. All right, I'm going to switch mics and we'll get going. All right, Shane.
Ready to go?
Yes.
Great. Hi, everyone. I'm Shane Lofgren, Head of Business Development and one of the co-founders of Manifold Bio. I'm going to tell you about what we do here. So at Manifold, our big goal is to both create drugs that help patients, as well as actually change the way that drug development is done, specifically by focusing on later stages of testing in vivo. So we have this process we call in vivo biologic design. I'm going to come back to it a little later, but I'm going to leave it up so you can think about what it might mean. So I'm going to focus first on the biologics part of this. So biologics are a large class of drugs that include antibodies. We're focused on a specific subset of biologics that we call target effectors. So target effectors are any type of drug that has basically 2 modules. One is an effector module that does something relevant in the biological system. The other is a targeting domain that drives that effect to a specific tissue, often disease tissue. So I'll draw those up right here. Yeah, this is the targeting arm of a target effector. And you have the effector arm. So common effectors might be toxins that cause cells to die. They might be immune modulators that drive a certain immune effect. There might be cells, if you're familiar with CAR-T. CAR-T is a type of target effector. And so the general property of effectors is they're generally very potent. If they're delivered systemically, they'll be so toxic that they can actually kill a patient. So you need that specific targeting in order to make these tractable. So the targeting domain's main purpose, as I said, is to drive targeting to disease. And if these drugs actually work exactly as I've drawn up just right here, where they only go to disease, they bring that effect just to disease, then actually I think we'd have treatment for a lot more diseases. A big challenge that comes up, though, is the fact that these targeting domains also end up targeting healthy tissue. This causes quite severe toxicity. Unfortunately, patients have actually died following treatment with these drugs. So a major challenge for target effectors, and specifically for targeting domains, is to figure out how to improve disease specificity. And that's what we're focused on at Manifold. So I won't talk too much about effectors anymore except to say that there are many off-patent, well-established effector domains that are available for us, for pretty much any other company to use. I will zoom in more now, though, on targeting domains and how we think about it. So zoom in like this. And so again, as I said, what we really want is disease specificity. We want more targeting, more accumulation in disease tissue. We want less targeting, less accumulation in healthy tissue. You can sort of convert those into one ratio, which is really what you're trying to maximize. And the key thing to know about disease specificity for targeting domains is that not every targeting domain, even against one target, behaves the same. In fact, there's quite a wide distribution of different levels of disease specificity for a given targeting strategy. This can happen because of different epitope. It's a different part of the target we're hitting. Off targets. These things are not perfectly specific. Biophysical properties. And finally, just format. There's been a lot of work recently to explore putting multiple different targeting domains together to try to create logic gates or another way to drive specificity. And so as we think about this whole space of parameters that targeting domain can have, this is what creates this distribution. And where we really want to be is right over here in this far fat tail at the end of this distribution. The problem is, though, if you just randomly sample, of course, where you're going to end up is somewhere right here. And so what current folks in industry do to try to get closer to that fat tail is use proxies, use in vitro tests, use in silico tests that have some correlation with the disease specificity we care about, try to move them to the right. But unfortunately, these proxies are that. They're only proxies. They're not especially predictive. And so when you actually go test your best things from your in vitro system in vivo, you often find they're still kind of mediocre. They're still kind of in this regime. So I'm going to say proxies get you here. We want to get all the way out here. And so we believe that to do this, the approach, there's kind of two key things you need to do. One is you need to get a lot more data from this distribution. And two, you need to bring more design where you learn what drives disease specificity from that data. And so that's what brings us over here to in vivo biologics design. So how do we do this? How do we get more data and how do we actually do design on that data? What we start with is a large pool of different targeting domains. So let's say this is targeting domain 1. Say this is targeting domain N. So we have a large pool of these. Now, what we'd like to do is mix them all together and actually stick them in a direct model of this disease specificity. The problem is when you mix all these together, you can't tell them apart once they're mixed. There's no obvious feature that can be easily read out. So what we created at Manifold is actually a barcoding system. So a small little protein tag that uniquely identifies each targeting domain. So this one gets barcode 1. This one would get barcode N. And what we're actually able to do, this is kind of the secret sauce of the company, is to actually convert these little peptide tags back to pieces of DNA, which can then be easily quantified on a linear sequencer. And so what this means then is we can actually take all these targeting domains, we can stick them in a mouse. It's my bad drawing of a mouse. We can also use other in vivo systems like non-human primates, which are also very good models of disease specificity. And then we can actually take the tissue back out of the mouse, quantify different tissues. And this gets us in vivo data. Very important. And we actually get distributions of properties in different tissues. We can find out how these targeting domains accumulate in the blood, how they accumulate in a tissue we care about, like the gut, how they accumulate in a tissue we care about, like a tumor. And what we're looking for, again, is molecules that have high accumulation in tumor, that retain well in the blood. That's really important. If your molecule is clearing too fast, it's never going to go anywhere where you need it to. And then critically, in this left side of the distribution in the gut, we want minimal accumulation in that healthy tissue. So hopefully we get there right in the first try. You know, doesn't always work that way, but that's where the design part comes in. And so we're actually able to learn from this data that we've generated and use that learning to drive the creation of the next batch, the next iteration of targeting domains, which we can then again barcode, because these barcodes are modular, they can go on any targeting domain, and run that loop again and again until we find that molecule that has the properties we want. I'm going to call it target star. And that is what becomes our drug. We have the targeting domain. We have the effector domain that we've written. There's lots of off-patent domains that we can use. It has low toxicity, which means we can dose it up until it actually works well and becomes an effective drug. And we're actually using this across a wide range of effector strategies, a wide range of targets and targeting strategies. And ultimately, though we're very focused on cancer internally, we believe this can be used across a large number of different diseases that we're tapping into with partners to bring the benefit of target effectors to, basically every known human disease that we think we can attract here. And so I just want to leave with one final note, since this is a conference focused on software, on data. So actually, as our founder Gleb likes to say, software and computation is the electricity that makes this whole process run. And so it's computation heavily informs the initial set of targeting domains that we focus on. We've computationally designed these barcodes using lessons learned from native proteins. The data, as you can imagine here, requires extensive computation for signal processing. And then finally, at the design step, we extensively use ML techniques, especially those optimized for lower end data sets, to enable us to more effectively design the next generation of target domains. And so we just raised a $40 million Series A. we're hiring a lot of folks right now to really build this out. We built the kind of the core foundation of this platform. We're using it to make our own drugs. We're trying to scale it up and make it better. So if this sounds interesting, let me know, or come look at our website. Thanks so much.
Definitely to ask you more about all of the last software 30 seconds at the end. We want to do questions, or we want to do questions at the end?
Let's do a few minutes of questions. People have them now, and then we'll move to the next speaker.
Okay. Anybody want to start? Yep.
How does the concentration of your molecules at the point that you're measuring relate to, or can you relate it to, say, the trough concentration that might be relevant for the therapeutic efficacy, or the maximum concentration that might be relevant for toxicity?
Yeah, great question. So
Can you just repeat the question?
Sure, yeah. So the question, if I understood correctly, is what concentration are we studying kind of these pools of molecules at, and is that kind of the same as the concentration that we affect the molecules to be both effective or cause toxicity?
Yeah, can you learn anything about the pharmacokinetics from your approach? Like, is the concentration that's going to be affecting the function, like the trough concentration or the maximum concentration in the case of toxicity, is that something that you can gain information about from your approach?
Well, what we can do is definitely dose these pools at different levels. And so if we kind of work backwards and think what's causing toxicity, you need a certain concentration of the molecule to drive effect. You can often learn this in vitro. And so we can basically infer how concentrated the molecules are in a given tissue at a given dose. And if we see that, basically we're dosing at some level and we're below that concentration in the tissue, I think we're using that as a signal that basically means we don't expect to see toxicity in the tissue. So basically, if we actually quantify what it means getting this fat tail, and that's below the effective concentration below EC50, we think that means that the molecule is less likely to be toxic.
Do you know that, say, for a given targeting molecule,
Yep,
it will still have the same tissue distribution without a barcode?
Yeah, great question. So the simple answer is not necessarily. So we actually have a whole process upstream where we select the most inert barcodes that we can still quantify. This is actually a key part of the work that's done on the platform. Sometimes, as you'd imagine, some of these peptides do drive biodistribution in certain tissues or do affect PK. So it's important that we screen this out. We also actually use multiple barcodes per construct, so we can average across them in order to really zoom in on what's driven by the targeting.
Really cool talk, and this is such a cool patient-centered design. I have a question about, do you count for age of the animals that you're using? I asked because I've seen some patients with the same tumor, is actually, they'll respond differently to based on age. So a lot of clinical trials, they'll enroll like younger patients because they think they have a better chance of making it, but they've actually found that older patients might respond to drugs, whereas younger patients may not.
So generally, no, not for the mouse studies, because I think mouse studies are generally done on mice at a very specific age. For non-human primate studies, though, we're able to learn a lot from non-naive. So basically, working with non-human primates that are where we don't have to kill the animal yet. And so there are colonies where the primates do span wide ranges of ages. So I think, and what we're looking for is a design that drives biodistribution kind of across the variation in the population. But we could learn if we see, we're seeing what we want when only in the older monkeys or only in the younger monkeys, but not all of them.
Selling the underlying platform itself versus taking away and selling it versus making a platform of battery distribution.
Yeah, it's a great question. We've actually spent a lot of time thinking about both business models. We really converged on the model of making our own drugs or making drugs for partners in certain domains that they're kind of more experts in. I think what really drives that is, basically, I think if you can make a drug, generally in biotech, especially with the macro situation the way it is now, it's generally better to do so. I think it makes your value more legible to downstream public market investors. And so for us, because these effector domains are so well established, it's actually not that hard. I mean, I don't want to trivialize it, it's still hard, but it's not that hard to actually take our target domains we've already made, compare them up with one of these, and have them on the drug.
So in your screening strategy, basically, it
iterate on the target, right?
That's right, yeah.
So how generalizable do those targets when you couple them to the vector domain?
Yeah, it's a great question. So the question is, what happens when you actually take this target domain and you couple it to the effector domain? So luckily, it's fairly generalizable, but we do have a final screening step where we actually couple the targeting domain with an inert effector domain. We don't want to actually kill the animal or cause other kind of downstream pharmacology when we couple those two. Just to confirm that kind of the effects, the biodistribution effects we're seeing with the targeting domain are maintained even when we have the effector domain.
So this is, I guess, a biology point that might pertain to your ML.
Yep.
Is the distribution in different tissues generally driven by the target you pick or the specificity for the target? Like, because, yeah.
Yeah, I mean, definitely for the target. So you can kind of imagine what we've already done. We've picked, like for our own internal pipeline, we have kind of our 3 leading targets that we're most excited about for cancer. If we swap those out for something else, it's going to drive wildly different biodistribution. But so even within the set of targeting domains that bind to a given target, there is still this distribution. And then that in turn is driven by these other factors here. So which region of the target are you binding? How specific is your binder to that target? Because these binders are never perfect. These are protein-protein interactions, as well as some of these other properties, biophysical properties like charge and hydrophobicity. That answer your question?
Partly, but I have to think a little bit more about how to phrase it.
Okay, sure. Yeah, happy to.
You guys will have time. We'll come back around kind of after everybody has gone. And I definitely want to ask you more about the software parts of your platform, because we do have a lot of folks who are coming at this from I'm an engineer, new to biology standpoint. But in the interest of time, I think we're going to rotate here to our next presenter. And yes, thank you very much. This was great. It's very brave to go first. So this is a great first talk. I would personally not have done it. So I'm very impressed.
Good whiteboard usage.
Yeah, you gotta plan your real estate.
Hi, good to see you all. I'm Nathan. I'm the co-founder of Ganymede. I will attempt to try and do as good whiteboard usage, but
I think I ask you to project a little louder.
yeah. So we're a software platform, pure software platform for integrating and automating your wet lab. And when I say your wet lab, I mean not liquid handlers or work cells, but rather everything: flow cytometry, bioreactors, assays, gels, et cetera, you name it. And I think for us, I'll go through the problem briefly, but the way that we do that is we build a virtual database over the things that we integrate. So we have a layer that's very good at integrating with the physical world, integrating with machinery, a layer that's good at integrating with apps and APIs and databases. And then we create a virtual environment where you can query and build databases and automation and run code on top of all of these integrations internals. And so the motivation for this, I think, is for us, biology is a very complex space. The structure of data in biotech, actually what was on the board before was a good demonstration of that. Think about the complexity of all of that ML data, all of the barcoding, all the in vivo stuff, all of the chemistry that needs to be tracked and related and put through systems here. Doing that and integrating that is very tough, and inevitably it requires effectively building your own systems in AWS. And that's the area that we want to tackle and help build very basic computing technologies and code and libraries for. So the way I think about biology as opposed to other domains that we've seen and that we have some experience in, an area like fintech or other more pure tech domains, oftentimes you build apps and you define your data models. And so here's your data model. You have some table with some columns, and this is related to some other table with some columns, and maybe you have another table, and that's about it. And there's lots and lots of data. And this applies even to an extent, I would say, to higher scale areas of biology, like comp bio, ML. You have lots of data. You have a finite number of schemas. You know what these are. There may be sequencing files or things like that. But at the wet lab level, that's probably the most complex domain out there of the complexity of the schema. And the data scale is not that huge. You may have at most tens or hundreds of thousands of records coming off something like a mass spec or flow cytometer. Oftentimes for plate-based data, it'll be hundreds or thousands of records. And that's where the issue is not the size and scale of the data, it's really the complexity of the schema. So you have all of these different tables that are all tightly interrelated, super highly relational, not super high scale data, and contextual to your process, contextual to your experiment. This is not necessarily stuff like a cell. People always want to come up with ontologies of what's going on in biology. And those sort of work sometimes, mostly don't. And that's not necessarily because this is data like, oh, a cell or a plate. It's because this is like your sample management or your process. Like, how would you say a hybridoma? How would you encode the process of actually doing that into a database? It's extremely complex. And so for Ganymede, that is our inspiration, saying, okay, this is an area where computing needs to be able to integrate very tightly with the physical world. You need to be able to really rapidly do business logic and really rapidly define schemas. This is part of the success of companies, I think, like Benchling as well, where they make it very easy to define data structures like that, define your schemas. And you need to be able to relate all of this and compute on it. So for us, I would say there's two parts of making this happen. One is how do you actually get the data off the instrument or off the app that you're integrating with. There are tools out there, there's companies out there for that. I think it's a relatively commoditized thing. So let's say you have your lab instrument. It's a big, I guess, pentagon now, rather than a box. This puts files on some local PC. Here's my file. And then, okay, you want to get into the cloud, great. This is doable. It's something that I think we do a fairly good job at and have good tooling around, but it's not anything crazy. But once you have this, once you have your file off your instrument, how are you going to actually get that into your LIMS system or your VLN? How are you going to get this into the context of your experiment? I'll give you an example of a bioreactor. So your bioreactor runs for several days. It generates a CSV at the end of that. Okay, great. Well, all right, what do you do with that? Well, you're going to do some analysis, but you need to attach a run ID to it. So okay, you're going to do some analysis, maybe outlier detection, aggregate it down, and then you will just know what VLN entry or what tracking entry you need to attach this to, and that will be how you attach this to your app, which I put in the cloud as well.
That looks like an egg.
I'm not very good at whiteboarding, as you might tell. So there's some in between getting the data from the file into the app. There's some, I'll make a gear, which looks like a sun. That process, there's two parts here. There's the mechanical physical connection to the instrument, and then there's this business logic connection of reshaping the data, analyzing the data, aggregating it down. And it's not very complex necessarily. You are probably just doing aggregation and then some pretty simple statistics on top of it, or like a curve fit. That's a pretty standard thing. But inevitably, there's apps have APIs and there's tools out there to get files off instruments, but you're still going to have to go into AWS and build your own thing to do that data reshaping and get the data from A to B. And that's where we provide that platform as well by creating our what we call flows. It's a series of tables and business logic in Python or SQL or what have you in between those tables, effectively like a database builder. And it's meant to evoke bioinformatics and computational biotooling very deliberately, because we want to bring that pipeline-like workflow system-like tooling that has been so revolutionary in bioinformatics and high-scale data to the wet lab level, and maybe so people can code on top of their wet lab data. And so that's where this notion of building a virtual database over your integrations, for us, the way that we do this, erase a little bit, is we say, okay, here's Ganymede. I've never drawn our logo in person. Like before. There you go. So here's Ganymede. We'll bidirectionally sync out this instrument table. We'll bidirectionally sync out the internal state of the app if we can. Try to get as much of this as we can. And then we provide the venue for writing code snippets, either as just standalone snippets or full Jupyter notebooks that can be pipelined together. For those of you who have used things like ML ops systems, that may sound familiar, very inspired by that, but intentionally detuned towards flexibility over performance. So I like to call it kind of a reverse Databricks. And so you can write your code here, and then we'll track the provenance through the code. So you may have your file coming off instrument, you write your driver in Ganymede, you write your driver, you write your analysis, you write your aggregations. right? Putting it into the app, all at the business logic level, and then we'll take care of the same thing, which is very, very unperformant. But again, it works at this lower scale data. And so we're not going to be running bioinformatic pipelines, but we might take the inputs or outputs of your pipelines and associate the run ID with them, and then put them in a folder structure, which is a super common problem that people have. Of, okay, well, where do I store my sequencing data? Or more commonly, where do I store my flow cytometry data or bioreactor? So, and we call this approach lattice code. The idea that it's not an API, it's actually virtualizing internals entirely and writing code on top of them. So, yeah, very inspired by things like MLOps, also inspired by things like the ETL revolution that's come to areas like product analytics in the last 5 or 10 years. You need to get the business logic out of production code and into data science scans and have engineering code at info level to support this. Our, I guess, market-wise, business model-wise, we have a usage-based pricing model for the platform itself to run the data in, we charge for record processing the end. And then we also do consultatively implement business logic too for people if they want that. And then we'll charge a separate license per and per integration type that we put into this. The market is pretty big. Hopefully we can capture, I like to say, all biopharma IT spend eventually. And yeah, AWS levels here. I think you guys can use really competing against AWS or in that sense. I would say our vision is, so this is a lab instrument. There's files coming off a lab instrument. There may be multiple files that are related together. It's really a schema that you're integrating, which is why you need business logic here. A CRO or a service provider is also just that. They are sending one or many files related together in a schema. You need to get it into your VLAN or lens. You need to maybe merge it with your data on your side. And so for us, there's never going to be a silver bullet to actually automate tech transfer and automate those files going around between companies. But we hope we can push it further rather than just, oh, here's PPI, we're reducing methodology, and we're almost a schema builder for transferring data between companies. So that's our dream. We are hiring as well. So engineers, scientists who can code, we need them all. So talk to me as well if you're interested in anything like that. And also happy to share anything we can around the structure and logistics of what we've been doing. We try to sort of open source our internals as much as we can, since for us, I think Unity is really altruistic endeavor to try and accelerate bio. And so happy to share whatever we can about the processes that come to the
Online question?
Yes, we do. Isaac's asked about dealing with multiple machines, vendors, platforms. Are you still able to find efficiency if you're doing idiosyncratic engineering for each platform, consumer, et cetera?
I would say no, and that's actually our core insight and thesis is that rather than trying to sell a library of connectors, we will be the place to write the connectors because there never will be a comprehensive library of connectors. There's too many, they're too contextual to what people are doing, like how you describe your sample management when you need to put it into your instrument data. It's just always going to be code. It's always going to have to be custom every time, and so at least we can ease the pain and be a good substrate for writing that code on top of it. So we'll try. We hope that this approach of putting everything at the tables and saying, okay, it's all tabular data, no JSON. I hate JSON because JSON abdicates the responsibility of structuring data. So if you do that, you're just kicking the can down the road. So future analysts have to build the JSON into a table so they can analyze it. So we do hope that that gives us an edge on capturing some commonalities about what people are doing. Like there are only so many ways to do flow cytometry, but that's not our selling point. Our selling point is that we build it and that we can build it efficiently.
So we've actually got a second online question too, and then we've got some here in person. So Nick has asked, does this require installing a driver on each instrument, on each instrument's underlying PC? Talk a little bit about what it looks like.
Yeah, so that's a function of what your IT team will allow, I would say ultimately. So ideally, so any drivers or business logic have to exist in the cloud. We will install an agent, a Windows, which is a very loose thing. It can be a Windows program ideally, because then we can not just get the files, but we can also associate, like we can see the instrument on the wire on the OS. But we may also install just a network drive configuration to keep the files up in the cloud and maybe build some metadata threading into the file structure itself, or sometimes even just an upload form. That may be all we can do based on IP policies. We kind of span the gamut from R&D, where stuff is much more permissible, up to manufacturing, where you are not allowed to touch anything for the most part. Any business logic has to exist in the cloud, and that allows us to thread provenance through the driver itself too, from a business logic perspective. But from a like getting the file perspective, yeah, we'll install something on the computer if we can.
Great. In-person question. You want to go I can't see your name tag, but you've got a kind of a green shirt. Yep, you. And then you here in the gray.
Thanks for the talk. How do you think about like the environment in which people get to run? Like restricting that environment or allowing people to have control of the environment where they ultimately like write these adapters? Like you mentioned Python, SQL. What is that like?
Yeah, so it's pretty open-ended. It's effectively as open-ended as Lambda. And in that sense, I would say it's kind of on users to maintain performance and maintain, to an extent, their own security, but we do have pretty locked down controls on what you can actually do. It's running in Airflow. We're effectively, it's a GCP Cloud Composer. They're managed Airflow, and then we've modified it a lot from there. But there's also a finite number. You kind of have to work with the state that Airflow gives you in that sense too. So we try to be arbitrary business logic, completely arbitrary business logic, because that's necessary. But then we're pretty locked down when it comes to like what packages we import and support. And so that's something that we oftentimes have to do, is bring in new packages that people need.
All right. You had a question?
Yeah.
Oh, yeah.
Thanks for the talk, Nathan.
Yeah.
I have a maybe a two-part product question. How did you go about soliciting feedback before on the product and the platform before you had any customers? And then related to that, how did you avoid getting biased by their particular preferences and use cases?
Yeah, I would say we've been able to grow pretty quickly, and a lot of that's because we took that bootstrapping approach of saying, okay, we're actually just going to consult for people, which I think is really the way to go. Like, do not come up with a product idea before you've actually implemented something with a client, which sounds topsy-turvy, but it's such a rich way of learning. And so from there, we abstracted it further and said, okay, now we're going to have more knowledge. First, before really having a product, we'll modularize the infra behind it, and then we'll start building a backend and some logic. And now we're about to roll out hopefully a public demo in the next few months, so that people will be able to be completely self-serve. That's the dream. But I think that's a really important way to go in an area this complex. And it's doable too. It was almost surprising for me because in my initial roles in tech, I was more on the B2C side in fintech. Then I migrated towards bio and enterprise over time. But you can really start with very little in the space. And I think that's a very effective way to do it. The bias, I think, is harder. That's more the art, I would say, of it, because it is hard. I think you just have to, I think you can't completely listen to people, because if you listen to people, you'll build like rift in their synthase, because the scientists don't want to code. And so they say, I have this complex process, I want no code, build me a no code workflow, like flowchart builder. So I think you do have to have a pretty strong opinion on like the technology a priori as well. I think that's important.
So once you've mapped all of this data into relational models, how does the data scientist or analyst get that back off? Is this like a web service interface or something that they're connected to, or how does that work with all of this now normalized data from various sources?
Can you repeat the question? Just yeah, the mics are on this end of the room, so anybody who has a question over there, just repeat it.
So the question is, how does the data scientist get the data back off once we've built all these relational models, and how do they use it? So the short answer is it's all in BigQuery. We're building, our database is actually a bunch of CSVs on the back end, and then you can query to BigQuery. We also have APIs and things like that. But I think the longer answer is we hope that they don't have to get it off, and that they can actually do the analysis in the ending too. If it's something super high scale, like yes, they'll probably have to set up like an ODBC connection to BigQuery, and then that's fine too. But we save everything. We want to have this be like a true backend builder. So rather than, I think a lot of workflow tools, there's the impression that, okay, this is kind of a copy of the data, you're doing an analysis, and then someone else is going to be the resting point source of truth. We want this to actually be a source of truth. And so we have a lot of versioning and a lot of controls around the data itself. So ideally, you don't have to get the data out again. You can just write more analysis in Gammy.
Then you got like a Python and R interpreter and all that, like kind of built in.
Sort of. We're building towards more of that being more natively integrated. Right now, we can spin up Jupyter notebooks dynamically that have the whole environment loaded up in them. So rather than saying, oh, I'm going to go from this table to this table with a single function, you can also kind of pull the whole web together at once with the Jupyter Notebook, where we give these tables. Eventually, that'll turn into an SDK, and then it'll be like, okay, now the Ganymede SDK can run anything.
Get it. There you go.
Sorry, one more question, I think. You get to browse him for an hour and a half.
Okay.
Yep, last question.
Okay,
Great volume.
you mentioned manufacturing data. Curious what your perspective is on GMP, GLP, et cetera, that kind of data that comes under regulatory scrutiny, where you need things like original sources of truth or certified copies and the like, and how your system might deal with that.
Yeah, so we actually came, I would say, more so from the manufacturing side. Our original, our first client was this company called Apprentice, their manufacturing execution system. So they work with big pharma for drug manufacturing. And so we've really been put through the ringer with that, I would say, on the robustness side. And that's where we save, because we save every single copy, it's very different than traditional IoT. We instead punt all the raw data that we see up to the cloud, and then are saving all the time, and then the driver comes after that. So it actually is pretty GMP by default, because we're just seeing every single thing we ever had come off the instrument. So there's cases where we can make instruments that are not GMP compliant, because we're saving every single signal that we see from them. I think for stuff like e-signature, we don't want to touch that. So we're more than happy to integrate with an ELN on the R&D side, or at the manufacturing side, like Apprentice, some SOP execution system runner. But in terms of actually saving the data, then it becomes for us more of a configuration thing, rather than our infrastructure by default should save everything. And that's another one of those things that's pretty unperformant. Like saving a copy of every version of the data you've seen doesn't work for billion record omics data, but it does work for like 10,000 record like scales, weighing and dispensing stuff.
Yeah, no, absolutely. Very cool.
Thank you.
All right.
Hi, my name is Kathy, and I lead the machine guided design team at Dino, and I'm really excited to tell you a little bit about what we do. I can try. Sorry. So, sorry, should I repeat that, or are we good?
This is Kathy from Dino.
Yeah. Hello. So at Dino, we're trying to solve the gene therapy delivery problem. As you may know, the human genome contains around 20,000 genes, and there are a lot of debilitating and or fatal diseases that are caused by having a non-functional copy. And so gene therapy is able to treat these diseases by supplementing the patient with a functional copy. And more often than not, we know exactly which gene is messed up and how to fix it. So the problem is, how do we get that gene into the patient cells? And one of the major breakthroughs in the gene therapy field was the discovery that AAV viruses are actually a great way to do this. So AAV stands for adeno-associated virus. There's a cartoon of one on my shirt. I'm going to represent it with a hexagon. It's a totally benign, naturally occurring set of viruses, and it has this like protein shell on the outside that we refer to as the capsid. And then on the inside, it generally carries its own genome. So the capsid is the part that's responsible for getting this virus into people's cells and delivering its genetic payload. So all you have to do is swap out the virus's genome with the gene that you want to deliver, and suddenly you have this package that delivers genes to the cells, which is great. And this is by far the most successful way of doing this, and AAVs are used in the only two currently available FDA-approved gene therapies. Despite a lot of the advantages of AAV, it does have two major drawbacks that prevent it from being more widely used. The first is that there are no naturally occurring variants of AAV that are specific for particular organs or tissues. So let's say you wanted to develop a gene therapy for the heart, and you give this patient a bunch of AAV with what you're target gene inside.
That's the best drawing so far, by the way.
So some of it will go into the heart, but a lot of it will go into non-heart tissues. So that means one, you need a pretty high dose to get enough into the heart to have an effect. And two, you're basically delivering a heart-specific gene to a bunch of non-heart tissues, and then that creates off-target effects and toxicity. So I guess that's one major drawback. The second drawback is because these AAVs are naturally occurring, they've been swirling around the human population for a while. So a large proportion of the population actually has pre-existing immunity against AAVs and are not eligible for gene therapy, up to half in some cases. So a lot of you have probably been exposed to AAVs without even knowing. So at Dino, we're trying to solve this problem by engineering better AAV capsids. And probably one, to be more specific, and two, to be different enough from naturally occurring capsids to evade this pre-existing immunity. We're purely focused on the delivery problem, so we don't actually make any complete drugs ourselves, but our business model is partnering with all of other companies that are working on the genetic payload, and thereby we can kind of enable gene therapies across the field for many different tissues and targets. Right now, we have actually 4 partnerships with 4 pharmaceutical companies and 4 organs. So I guess the reason this is hard is because this capsid is made up of a protein sequence that is around 735 amino acids long. You can think of this as a string that's 735 long and with an alphabet of 20. This thing kind of folds into a protein, and 60 of these come together to self-assemble into this capsid. And if you make just a single change to one of these letters, you have about a 2/3 chance of destroying its ability to even fold into a capsid, let alone target a specific tissue. And on the other hand, with a length of 375, the search space is very large. So you have both a vast search space and very sparsely populated with functions. And so this is kind of a great place to try to apply machine learning methods that I know. So these methods are generally very data hungry. So the way that our platform works is that we can generate many different variants of this protein on the scale of hundreds of thousands. And we can assay them for their ability to fold into capsids. And we can also then test them for their tissue specificity in several different contexts, including non-human primates, which is our primary context that we're testing in, as well as mouse and also human cells. And the reason to test in multiple contexts is that if it works in all of them, then it's more likely to translate. I do not have good board management, so I'll put that here. This generates basically a very large amount of data. You can imagine this is 100,000 rows, hundreds-ish columns, and our platform is actually currently generating about 1 billion capsid measurements a month, because our methods are very data. And from here, called box data. From here, we then use this to train neural models, and it's starting to look very similar to Shane's presentation. I'm regretting going third now, but and because we have so much data, we can really employ a lot of these deep learning methods that are successful in other contexts. And we can use kind of a variety of different methods because they have different effective biases and are successful in different contexts. And from there, we can use this to generate new sequences that we think will. And because we're using models that represent this sequence to function relationship in silico, we can do a lot of the screening in a computer, but we can also directly generate sequences by, for example, sampling a generative model, or we can, if we have a model that is differentiable, we can do gradient ascent and those kinds of methods that are much more efficient than just trying to randomly change the sequence. And from there, we can feed these back into our experimental pipeline and have this multi-round optimization loop. And I guess the benefits of our platform is we can directly encode all of the variants that we want to make and measure them all individually, which is great. And we can do this at a scale that enables a lot of these methods. I would say some of the advantages of China's approach are that, one, we have a very focused approach. We're only trying to solve AAV gene therapy delivery. And this lets us benefit from a very specialized experimental pipeline that can bring out very relevant data for our problem, and also a lot of domain-specific expertise when we're building the models. And the reason that's very important is because, unlike in a lot of other ML fields where you're trying to generate new things, like here's a bunch of faces, give me a face, or here's a bunch of cats, give me another cat. This is like, here's a bunch of capsids, give me one that's better than anything you've ever seen before. And kind of being successful at that means giving your model a lot of good priors and regularizers, and that requires a lot of domain-specific knowledge. Yeah, so be very focused, but at the same time have a broad impact in the field because we are a platform company and our technology kind of enables gene therapies across many different organs and diseases. So this combination of being able to be focused, but with the broad impact that I think is kind of unique and one of the things I really like about working with China. Yeah, so we are also hiring software engineers, ML engineers, ML scientists, data scientists, all those kind of people. And yeah, I'd be happy to take any questions.
So this is really cool. In the first step where you're creating variants, is this hypothesis generating or hypothesis testing? What I mean by that is, are you going in with all of these variants with hypotheses around specific sort of mutageneses or like amino acid substitutions you were thinking will confer greater specificity, say to the liver, to the heart, et cetera? Or are you completely agnostic? You don't know, and you just wanted to do generate the hypotheses as the experiments go through. And similarly for like immunogenicity, are you going in and making these variants with the hypothesis in mind, or are you allowing the loop to create hypotheses for you as to how certain variants might map to certain components of like the underlying causes of like immunogenicity?
Yeah, that's a great question. So I would say a little bit of both. So in some cases, we have some prior biological information that we can use to generate sequences that may be higher likelihood to work. But also because we can design these variants, we can actually design data sets that are good for machine learning models. So ones that span the space well, maybe using some ideas from sensing, for example, to actually build models that are going to be maximally useful there. So we'll aim both for biology and for better models.
Great talk. And so my question is about, I guess, off-target toxicity. How are you assessing every tissue where you kind of might have off-target effects? And then second part of my question, are you kind of taking that data and maybe penalizing some of your models to kind of account for certain sequences that then might later lead to off-target effects?
Yeah, so the answer is yes to both of the things. We do collect every tissue in all of these experiments. And we have kind of what we call a capsid profile when we're actually optimizing for new capsids, which is a wish list from people on the business side. If I want a capsid that targets the brain and detargets the liver and detargets the spleen. And so we're definitely weighting those properties accordingly.
In the white tank top.
This is great, such cool tech. But I have a question about variability between species, because I remember some of the really early mouse brain specific data, you could see huge amounts of variability from mouse strain to mouse strain. I remember like it would work great in black sixes, but like not super age, something like that. What is your, like, are you seeing that same thing? And is the way built in non-human primates like to account for that? And sort of like what is the hit rate that you get from species to species compared to like what's been seen?
I can't say exactly what we're seeing differently, but we are seeing definitely differences in the brain is definitely a place where you see a lot of those differences. So one, we're using non-human primates because we're hoping they're a little closer to humans than mice. And also we're using multiple species because it is kind of conserved across several mammals. Maybe there's a big chance to work with humans, but of course these things would have to be validated.
Sorry, quick follow-up. Do you have to also then I assume that cross species validation means you also likely have more promiscuity. Like, how do you account for like broad promiscuity? And as you say, it's going to hit the human cell, but not the mouse, or hit across multiple organs. Does that also just make it less specific?
Sorry, can you say that again? What
Sorry,
did you
I guess my question is, by selecting for AAVs that broadly target across species, does that also increase the lack of specificity for organs?
I think that it does make it a harder optimization problem, but if it increases the likelihood of later success, then I think it'd be worth it. So there's like a trade-off.
Oh, great. I was wondering about, I mean, I think the good news is that you only need to find a few for the collaborations. It's not like you need to solve, you need to get millions, but it is, the bad news is that the space is, right, it's bigger than the length to the 20 because you've got indels. So you're doing this cycling in order to optimize and I suppose I wondered if there was anything that you could share about how you narrow down. You talk about 100,000, right, as if it was a large number, and it's sort of like 10 to the 11, 12 smaller, conservative estimate of the space. Because you've got quite a lot of experience now with the company, and I think the world is very interesting.
Yeah, so I can give you a should I repeat that?
Yes, please.
So the question is, the space is very large. How do you narrow down to 100,000? Next experiment. So I'll give a brief answer, but also we gave a talk about this at a excited gene cell therapy conference that also goes into methods more specifically. But what we do is generate on the order of billions of potential candidates in silico. We score them with our models on wide range of different models on both what we think their property is going to be, as well as our uncertainty on that estimate. And then we kind of have this like global portfolio optimization algorithm to build the final set of 100,000 that kind of accounts for maximizing properties, like accounting for diversity and also accounting for uncertainty. So trying to really hedge uncertainty, just kind of want to hedge.
All right, let's save remaining questions for the end, just in the interest of time, but we will have plenty of time. And we will move to our last presenter here. Aaron now has to come up with something that's better than the stick man with the heart, and I don't know that that's possible.
It's a final speak if everyone's still awake. I mean,
That's true. If everybody's still alive, then you've already won. But Kathy kept him up.
Brilliant. Well, fantastic to see everyone. My name is Aaron. I'm the CEO of a company called Postera. And I guess what I'd love to do today is start incredibly high level and eventually continue to drill further and further until Lizzie tells me my time is up. In short, my company focuses on medicinal chemistry powered by machine learning. And what you've heard already is a very kind of broad spectrum of a variety of kind of therapeutic modalities, if you will, for how we cure diseases today. So you've heard both from like Dino and Manifold about more like cool cutting edge gene therapy or protein therapeutics or biologics in general. And you can think about how the FDA approves drugs between traditional SM, small molecules, and then all of the kind of biologics space, gene therapy, cell therapy, protein therapeutics, antibodies, etc. Now, to date, small molecules, depending what year, look, is somewhere around like 70% of all approved drugs. And then you've got this 30%, which is growing super fast over here. So just to start with the context, Postera is focused entirely in the good old fashioned small molecule space. And so I'm going to focus entirely on what this type of process looks like in order to bring a cure to patients. And the way we think about it at Postera is effectively a three-step process that takes a long time. You effectively start out with biology, you move to chemistry, and then effectively you move to medicine. The idea here is I want to focus on a given disease, say Alzheimer's.
And my starting point is, what is the fundamental biological mechanism that causes Alzheimer's? And therefore, what is the fundamental biological target that I need to go after in order to ameliorate the effects of this disease? That is effectively forming the problem, like what is broken in the body that needs to be fixed. Chemistry is then your solution in the small molecule space. How do I design a therapeutic that can be safely and efficaciously administered to a patient in order to resolve the biological problem? Medicine then becomes, who is the correct set of patients that I can actually administer this drug to? And that is the whole process of clinical trials, phase 1, phase 2, phase 3. Now, in general, these guys here are referred to as the preclinical stage, and this guy is referred to as the clinical stage. In terms of a breakdown, this whole thing, roughly, depending what stats you look at, between 8 to 10 years, $1 billion to $2 billion. It's really expensive. It takes a long time. It's not really getting that much better. Now, within this space, which is where Posterior focuses, and specifically on the chemistry, this is around 5 years, so about half the time taken to develop a drug all the way through. So I'm going to focus and narrow in on the kind of chemistry portion, which has its own like sub-process, if you will. Medicinal chemistry is effectively just chemistry that's used for medicine. That's really all it is. Now, chemistry or medicinal chemistry itself can be broken down into yet another 3-step process, which is design, make, and test. Very simple idea, and this is a cyclical process, not a chronological one. You basically design a molecule. You've then got to make that molecule, and then you're going to test that molecule in an assay, and you're going to try and figure out, does this thing look promising such that one day I have enough confidence to put it into a human body. Now, within the kind of AI for drug discovery space, within the early days, a huge amount of time and attention was focused on design. And there's a couple of reasons for that. One, it's just kind of quite sexy to get an algorithm to dream up molecules. Like that feels like a cool feat to work on. Secondly, the actual ML metrics and benchmarks are much more clean in terms of how I determine whether I have made a vector molecule. Namely, has the potency gone up? Has metabolic stability improved? Has the solubility gone down? I have like concrete metrics that I can see. Really, Posterior's kind of, I guess, value out of what we're trying to bring to the industry is that for the first time, we really unify the entire design, make, test process with a lot of the pioneer machine learning work that our co-founders have done. You can read many of our publications on the site. But really what I want to focus on for however long I've got left is actually this middle stage here of how to make molecules. The reason I want to focus on this is not only because the ML is much more challenging, but it's actually the majority of the time of this cycle. These are not evenly distributed. You typically are seeing this take about 10%, this take about 70%, and this take about 20% in terms of your time. So your bottleneck to doing chemistry very well, that 5-year horizon I talked about, ultimately getting drugs into clinical trials, this guy is your pain point, and this guy is where you really want to invest a lot of the time and energy. And that was quite a paradigm shift when we went to investors, because everyone always wants to talk about design me a better molecule. And our argument is that actually this guy is where you really want to focus on efforts. So within the process of kind of making molecules, what we're talking about here is a process known as chemical synthesis, or more specifically retrosynthesis. The idea is if I have some target molecule that I want to make, that ultimately I want to assess whether it's good or viable and useful, I fundamentally have to start breaking it into pieces. You can kind of start imagining this like almost chemical Lego. You start out with very simple building blocks, and I ideally combine them in such a way to get to the target molecule that you want. Now, why is this a notoriously difficult problem to try and solve? Firstly, it's got 3 components, and I'm going to come back to these 3 components in a second, and I'm going to put really bizarre acronyms that won't make anything, won't make much sense just yet. But you really need to do 3 things in order to do retrosynthesis really, really well. It's also a very nasty problem for ML because it's not a a many-to-one problem. It's a one-to-many problem. There are many viable ways that you can make this target molecule. And so having really rigorous benchmarks is pretty much subjective. And one chemist will give you an entirely different view as to an optimal way to make a molecule. It's not nice and clean like often the design process. So it's kind of hard to figure out whether you're making any progress at all. What makes this even more difficult is that depending on your starting materials, you can access certain parts of chemistry that you wouldn't with another set of starting materials. So again, it makes it hard to compare one group with another. And so what Posterior has really done over the last 2 years is effectively try and cover these 3 parts of retrosynthesis. This stands for building blocks. What are the starting raw materials that I have at my disposal in order to get to this beautiful dream molecule I have over here? The world we live in right now is quite amazing in that if you wind the clock back 10 years, it was really big pharma, Pfizer, Novartis, etc., who had the biggest inventory of building blocks and thus had the most likely chance of finding some novel chemistry. That is no longer the case. Today, there are well over 20 billion materials that you can buy from labs around the world, either on the shelf or within like a week or 2. And that just means, imagine, let's say there are about 50 workhorse chemical reactions with over 20 billion starting materials. You can imagine the combinatorics become pretty intimidating pretty quickly. And so the first challenge is, how do I aggregate and get a dynamic view as to what are the starting materials? So Posterior is integrated with pretty much all the major world CROs so that we now know their inventory better than they do. And on a monthly basis, we have a good idea. What materials do you have? We actually know what chemistry is working on what set of materials so we can allocate work resources. Very logistics heavy, but really critical. Now, the second part of the problem is these lines. This is a reaction. How do I predict whether a reaction is going to work? Because if you fail, that there is no way to kind of resurrect it. You've lost everything. You don't, it's not like you get 10% of the way there typically. And so one big part of what Posterior focused on was given inputs, how do I get an output? We use a kind of representation of chemistry called Smiles notation, which is effectively at looking at language. And we trained a transformer architecture very similar to the kind of Google publications coming out of Brain around like transformer usage. And basically showed for the first time in a paper back in 2018 that we could compete with some of the best chemists in the world at predicting the outcomes of chemical reactions. That doesn't solve the whole problem. You've still got to deal with these other pieces, but it was a real milestone in the industry for being able to say that algorithms can begin to have good chemical intuition about what is tractable in chemical synthesis. The final part is that because this is a one-to-many problem, you have many viable routes down this graph. It does become a graph search problem. It's really foolish to do a very greedy search and just prioritize the most practical reaction or the one that's had most precedent in the literature. So you end up with a huge graph search. And so honestly, there is a huge amount. I view this as a logistics challenge. This is an ML challenge, and this is a pure engineering challenge. And we probably spend 6 months getting all the inventory, 3 months building the model, and a year and a half doing the engineering. When we first built the, I guess, original version of our retrosynthesis model, an individual solution to this problem would take around like 15 minutes to find a single route. That's now down to about 5 seconds over the last 18 months. The upshot being, when you put synthesis first so that every molecule you design is by definition already makeable, not just makeable, you know exactly which CRO in the world can make it, it really does increase the speed at which you can get more data back from your experiments, make better models to design the next round of synthesis. The upshot is Posterior is now doing that 5-year side plan around 18 months, and we want to keep improving. That's not just in theory, like we've actually done this in 18 months. We work with Pfizer, we work with the NIH and several others who we're now partnering with to work on drug discovery programs, as well as a bunch of stuff we do internally. So much more to be said. I'll pause there and take any questions, but we love synthesis, we love drug discovery, and we love physical chemistry.
Questions? Yeah.
So when you're looking at the records problem, how have you guys found like a lot of these like pre-trained style models compared to like getting our literature?
Can you repeat the question?
Yeah, he was, so the question was, how do we find some of like the pre-trained, I believe, language models as opposed to more, I guess, alternative representations of chemistry, for example, like using graph transformers. We were quite surprised because SMILES is a one-dimensional representation of chemistry. Graph is at least two-dimensional. We have a kind of like all the other tens of millions of reactions. We actually find the SMILES representation works just as well, if not better than graph transformer models. I don't have an intuitive answer as to necessarily why. I just think at scale, that one-dimensional representation just eventually is no less significant than a two-dimensional representation as long as you have sufficient data.
I just would follow with your inference time fast those models are curious. Like is running inference on using these small transformers like faster or retraining faster than say retraining a graph transformers?
Good question. I have to get the stats on you for that. The inference for the transformers is naturally challenging just because you've got the beam search at the end. And so a lot of what we did was just converting Python to Rust, trying to get this thing super, super fast. But I don't actually have the times of transformers versus graph transformers, but I'm trying to look at that. Right now, it's not really the bottleneck. And you actually don't need that much pre-training. We do a huge amount of pre-training for the design and test part of our platform. The make actually, we found it to be okay, even without. Yeah.
How often do you find that your predicted syntheses just don't work, and what do you do with the failure? And maybe the associated question is, anybody who's talked to a medicinal chemist in pharma knows that they're quite an opinionated bunch. So how do you get them to buy into it?
You know, it's a fantastic question. So to your first point, absolutely. So over the last year, we have made thousands of molecules, and our synthesis success rate is about 93%. Industry averages are about 60-70. So there's still 7% that we fail at. And that's valuable. Like any CRO that we do synthesis with, our contractual requirement is you have to give us the failed reactions. That's the problem with a lot of public literature on reaction chemistry. People only report the good stuff, right? No medicinal chemist or synthetic chemist says, I tried this 3 times and it didn't work, so I changed course. In terms of like medicinal chemists, long discussion here on culture fit. And it's something we index very heavily. I think from my perspective, what our medicinal chemists really value is that not only do I have the capacity to see 10 billion building blocks, which I could never memorize in my own head, and often it's not like our retrosynthesis will ever propose a reaction that they've never thought of, but very frequently they will see a building block that they never knew was there. And the upshot being what used to be a 3-step synthesis 2 years ago is now a 1-step synthesis because of the existence of some like complex intermediates on some CRO shelf in the back end of Ukraine. And that is where you save the time and the effort in the cycles. Okay.
So, I mean, objectively, also kind of a med chemist point, like building block chemistry, med chemists, and process chemists will all focus on entirely the references. So like, how do you remedy the differences between the data sets that are literally operating in different sequences?
It's a great question. The honest answer is our existing kind of platform today is very medicinal chemistry heavy, not process chemistry. In medicinal chemistry, you really care about rapid, fast cycle time. And if my yield is like 70%, like whatever, deal with it. I've got enough for my assay, let's go. Process chem, obviously, you care much more about optimizing each step of the reaction, and really yield prediction, which is a notoriously challenging problem. It becomes the kind of like gold star. It's still a work in progress. I don't really think we've made huge leaps and bounds in that area, but it's certainly something on the
But like even building block chemistry will often emphasize like water soluble chemistry, which is not even something that a med chemist would think about, right? Because you need to scale rather than even. So.
Yeah, I think for us ultimately it's really lead time and cost that drive the decision making. Because to us it's just getting data back as fast as we can. So I don't have the breakdown of like how much water soluble versus non we do in our data sets, but I think whenever our algorithms, which typically have tens of thousands of viable routes, we only send back about 10 to the chemists. It's really prioritized for speed, reaction reliability. Ideally, it's got some precedent in the literature so the med chemist doesn't panic, and really just like good, reliable building blocks that we know are actually there on the shelf of the CRO.
One more in the back.
Can you comment on the graph construction, specifically for the type of organic chemistry reaction which are possible? How did you go about assembling them? Did you talk to each CRO and understood what type of reaction they were comfortable with? Or did you talk with medicinal chemists and ask them what type of transformation they were comfortable? Can you comment a bit more on that?
So the reactions that we use are actually learned from data. We don't go to a CRO. Okay, slight caveat. The starting point was not going to CROs to say, give us your best reactions, and then we'll formulate our routes. What we went to them was, give us the building blocks, and our kind of data set of reactions will figure out how to get your building blocks to the end molecule. Now, subsequently, what we've done is we've realized that actually some CROs are more specialized in some chemistry than others. And now we actually have a lot of their proprietary data on their success rates. And we will redirect actually the actual orders depending on whether we think this type of chemistry is amenable for your chemists. But it wasn't the starting point. It's helpful, it's not necessary.
Right, I'm gonna call it there. Let's give a hand. All right, so all that's now between us and
We have lost our battery. All right, hang on. Keep talking.
I lost our battery on the camera.
Yeah,
That was great timing for that part.
it was really good
Do we have
timing.
a spare battery?
Hey, that's a.
Okay,
All right,
so
we're
hold
on Peter's laptop now.
So Peter, we're gonna switch to the Zoom camera on your laptop, which you can position on the chair. If you move your laptop there, sit there. We're gonna take 4 chairs, turn them around. We're gonna do this in we'll do it in a pretty quick panel because Liz just brought up all the food. And I'm sure the food and the drink are just as exciting, maybe more so. If we get the 4 speakers, actually we're doing 1 substitution, I believe, for Ganymede. Is that right? All right, come on up.
All right.
Can you introduce yourself for your team?
Oh, yeah. Yeah, so I
See
didn't
up your
talk.
nose.
I'm Sammy. I'm a founding engineer at Ganymede. So filling in to answer some of the more engineering heavy questions.
All right.
So just given the theme with Bits Bio at the sort of intersection of software and bio and biotech, a little bit of ML, we thought we would ask a few more questions that are directed in that vein, and then open it up for 1 or 2 more from the audience. You're gonna make them sit?
Yeah, we're looking at your waist.
Oh, well, I don't need to be on camera. That's the point. These guys are the.
All right.
Yeah.
All right.
But I'm sure I'm very loud. So, all right. Great. So the first question we thought we'd ask is just very straightforward one. Briefly, in each of your companies, are there any aspects of software engineering that you guys do that requires the development of new software versus, say, off-the-shelf products? And how do software engineers make a difference in your organization? So we'll just go like left to right.
Sure. So generally, no, we're not doing our own software development. We do a lot of pipeline development. So we have 2 computational biology FTEs. We don't have any software engineers. That said, me, Love, and Pierce all have computational backgrounds. Love used to be a software engineer at Google. He actually built a lot of our initial AWS infrastructure when we first started out. So I think that perhaps we had a different set of founders, maybe we would need to do some different software engineering, but generally off-the-shelf stuff.
So if you broaden that a bit to computational biology, machine learning, and so on, is the answer. So you said you are hiring in that direction.
Yes, for sure. Yeah, we're definitely hiring computational biologists. We do a lot of pipeline development. We do a lot of development of our own signal processing algorithms, a lot of development of our own machine learning model. I mean, we're inspired by already established machine learning models, but then we customize them for our challenge.
Yeah, we're fortunate in the sense that we have a lot of building blocks, I guess different use of building blocks, but like infrastructure as code that we're using. So we have a lot of things in place where we're able to just configure it to the platform that we're building. Oh yeah, sure. And we're also going in the opposite direction, as Nathan mentioned, in trying to build abstract platform where people can write their own connectors to lab automation equipment and instruments. So we're coming at it from both directions to develop the platform. We're fortunate in the sense that a lot of the cloud infrastructure is already in place where we can build up existing infrastructure and solutions. And then coming from the other direction, we're trying to stress test a lot of different use cases and building connectors to make sure that we're meeting in the middle. So we have a team of 4 software engineers, and we're also hiring software engineers. Yeah, a lot of what we do is, I guess, a mix of configuration and also building from scratch. Probably half and half.
At Dyno, we employ about 7 software engineers, and then maybe another 2 dozen of people who cloud provider and our Argo pipelines on top of that, but after that, we're kind of building our own pipelines and ML are those little specialized, and we haven't really found any tools that work well for that. So we're building our own data processing.
Yeah, I guess opposed to get to meet one of our engineers and just say hi to Jonathan at the end. We have a team of about 8 engineers and a team of about 6 ML folks. Pretty much everything is custom within Postero, particularly ML. A lot of the engineering, we do rely very, very heavily on AWS, but I think for us at Postero, it was the, I guess, technology foundation first, and then the chemistry coming after that. And so medicinal chemists and domain experts coming into Postero are actually being indexed to fit in with an existing software culture rather than necessarily the way around. And so wherever we can leverage it, we love some open source. There's a phenomenal kind of package called RDK that we use MLflow and others, but we certainly try to avoid any reliance on like third-party commercial software.
Awesome. So next question is actually about open source. We were curious to know, with regard to either code or data or open publications about methods, does your company have a stance? Have you done it before? Do you intend to do it? And how do you just think about that world in relation to the value proposition? And we'll go the other direction.
Sure. I think we've actually done one step further this year, which is we open source drugs. We actually launched an initiative called COVID Moonshot, which has become the world's largest open science initiative to develop COVID antivirals. This has basically gone from our founders just throwing up a website in March 2020 to now a drug getting ready for phase 1 trials with no patent owner, no profit. And our hope is that not only can all of the subsequent data generated as part of our technology help the rest of the world for any future pandemics, but that actually for a lot of the developing nations who won't be able to access vaccines until at least another 2 years, and for whom Paxlovid and Pfizer's drugs are phenomenal but still expensive, we can offer a super dirt cheap alternative that is incredibly effective. And so actually in Postero, we've tried this idea of open sourcing drugs, which will not work for every disease by any means, but this next year should be the first time in history that a drug has gone into human trials with no IP attached to it. So super proud of that effort, and we hope a lot of the work we've done can help a lot of the researchers with building better chemistry tools.
I think as far as open sourcing models or data, that doesn't really make a lot of sense for a business model at the moment, but we might consider it later if our business model shifts. As far as software, we have actually open sourced a piece of software, which is custom SDK for Argo. It's called Hera. You guys should check it out. So I think we're definitely a lot more open on open sourcing software, especially tools on these workflows that we build.
And publications.
And yeah, also.
You guys have an amazing page with like a bunch of.
I guess that's one form of open sourcing, which is a lot of publications and content.
Yeah, so for Ganymede, open source is really important because it lets people build on top of our platform instead of just using our platform. And similarly, we're trying as a platform to expose any open source packages or things that might be useful to different audiences, that we make it accessible to them so that they know it exists. We're also working on, I guess, some things that are new in terms of platform as a service. So something like in-browser, code editor, IDE that we'll plan to open source down the line. Yeah, also just in terms of using existing packages on top of Ganymede is super important.
Yeah, I'd say for us, we do feel like the know-how, the methods, the data pipelines are pretty important, very important for our differentiation right now in a pretty crowded therapeutic space. So we want to be realistic about the impact on our business if we share too much too soon. I think though we do have a roadmap of getting more out there. I think near at hand we'll be publishing the underlying method, how we actually make the barcodes, how we quantify them, sequencing. So you can expect to see that potentially in the next few years. Eventually, I think we're open to the idea of putting more of the data we're generating out there to help people learn about the underlying properties driving by distribution targeting for biologics. And I think, like I said, we're an open mind, but for now, we are keeping our cards pretty close.
Okay, so let's go for about five more minutes or so we can get some food. And I think, you know, you can direct a question if you have a question for a particular person, or if you just like anyone to speak up to your question. Both are good. Yeah.
You're all looking for software engineers. What's the one primary skill set that you're having a hard time finding?
Anybody?
I would say for computational biologists, which is more of our focus for now, we're most keen on biology intuition. So oftentimes, one way, not the only way this happens, is when you have somebody who starts with a foundation in biology and then has made a move over to computation. Because it is very important when you're analyzing our data, it's very specific, and you're also contributing to experimental design, which is also something that computational biologists do, that you are able to think like a biologist. So yeah, that's important.
Yeah, for us, we're looking for people who can operate well in very ambiguous situations. And we're really early stage startup, and so that means we're in a lot of different hats, especially at the size that we're in now. And so yeah, that's what we're looking for is people who are strong software engineers who are able to operate in a lot of different environments, especially when you don't anticipate what those environments look like.
Similar to the other answers, we're looking for people who come with a lot of expertise in like systems design, for example, or fields that they're very comfortable with, but are able to really understand the problem and the experimental pipeline that we have and the goals that we have, and kind of bridge that communication gap.
Yeah, I definitely echo the kind of dearth of domain knowledge for very, very kind of qualified software engineers, who kind of why I love this community. I think an additional nuance as well is your typical software engineer has probably been, we like having industry experience in first year, they've probably been a tech company or maybe like a software as a service company. And if you think about like their incentive structure or how they know they're doing a good job is revenue went up, right? Like I rolled out a feature, clients liked it, and now the ARR of my company has gone up. And it's much easier to tie the work of an engineer to the top line revenue growth of a company. In a biotech like Osteo, where we measure our progress on how rapidly can we get a drug into like the clinic, it's harder to find, I guess, engineers who can maintain that level of like just, I guess, firstly, high level, big vision picture of I know why I'm rolling this feature out, even if I don't see its impact for maybe like a couple of years. And so I think really finding engineers who are so curious that they will go sit in the medicinal chemistry meeting because they want to see how the tools that I've been building these last couple of weeks are actually feeding into your workflow, because that's for them where the incentive originates, as opposed to revenue went up last month. I think that's a shift psychological.
Actually, building on that question, one thing that people have heard a lot, so you hear a lot of, hey, all of these new computational tools, all of this new engineering tools, bringing software to biology, we can unlock a ton of value. You also hear the flip side, wherein that's hype, that's fluff, there's no real value here. How would you guys react to the latter statement? There's always less value than, you know, some marketing copy might make it seem, but how would you react to people who say, really great in theory, but in practice, we're going to do this traditionally?
Yeah,
I stumped the panel.
Yeah, I can start with this one. So yeah, something that I've seen as an engineer just joining, you know, like meetings with a lot of these top biotechs is it's fascinating to see IT or software or data leaders at each of these companies articulate the same exact problems without communicating with the other biotechs. And so we have the advantage of being in all these meetings and talking to all these different companies that articulate the same problem. And that problem, I think, is something, again, that we've already identified and are working towards solving. So it's really impactful being at a place where we can talk to these companies, see the same problem, and then address the problem, and hopefully down the line see our solution be the right one.
Say for us, so me, Glove, and Pierce all have backgrounds in ML. We've all got very excited by the potential of applying. It's a very powerful tool to biology. I think for us, we do see it as very powerful, but I think there's a big bottleneck in terms of the data generation side. So that was a key motivation for Manifold, was to create this technology that would let us test more than just design in vivo, a lot like Dino has done. And so I guess to cynics who say ML has not a lot of value to add to biology, I think still need to, I mean, obviously we need to see what it can do, what it has, sufficient data like it's had in more classic tech domains. But I think we feel pretty optimistic that with the right data, we can go on with it.
Great, another question then? Peter.
My question would relate to a question that was asked earlier. It's about an idea that is common in machine learning, especially reinforcement learning. It's like these exploration, exploitation trade-off. So a lot of I think a common pattern in at least three of you is in your companies is that you use machine learning to boil down a large search space. You have like a massive search space, and you want to iterate the process of optimizing on that. And how do you, for example, for Dino, you have a massive space of potential bioreactors, right? So how do you don't fall into the trap that you picked a few viruses at the start and just optimize on them, but they've got the wrong guess from the get-go. So how do you balance this trade-off? How do you, when you decide to explore more, or when you decide to exploit what you already learned from the data?
Take that one. So I think that's a really good question. And I guess the first step is recognizing that this trade-off is important and also being able to quantify it and formulate it as an optimization problem. So like even thinking in these terms, I think gives you a real advantage because you can quantify stuff like sequence diversity. So I think it's about how you can directly tie those things to the business value and the horizon that which you want to achieve that business value, and then kind of work off of that.
Add to that, but this is really important. Just like the practical consequence of this is if you take a very exploitative approach very early on. Within our domain, you can end up chasing a few kind of like important early, I guess, properties of molecules. Like it needs to be super potent, and everyone gets very excited about how potent my molecule is. It's rarely the rate limiting step. By the time you've got a super potent molecule, you have effectively barred yourself into such a narrow chemical space that when later on you need to optimize 6 other properties to get decent in vivo PK, like you just can't get out of that like small chemical environment. And so within Posterior, a lot of work around like Bayesian and ML and having good and principled uncertainty estimates, I guess, is the kind of technology answer. There's a couple of publications on our website about that. The practical answer is when we do partnerships with Pfizer or with others, we stipulate very clearly that you need to give posterior autonomy for your benefit. Because if you don't, if you involve too early and you try and cherry pick from the algorithm's designs, a long-term strategy of I will suffer some weaker compounds now to make more informed decisions down the line. If you reduce the algorithm's ability to do that, we think you're more likely to get stuck in this more classical like local minimum chemical space. So I think the upshot is it allows us to be more principally argued about autonomy within like BD partnerships.
Right. I think that was the perfect last question because we've done a lot of exploration and you guys can exploit specific questions. So another round of applause for thank you so quick. Huge, I mean, Liz, everything you see here, logistically, I mean, amazing.
I literally built the building.
So. You built it. So big, huge thanks to Lizzie for everything. Videographer Peter here was spent half the weekend figuring out how to stream and connect cameras we stole from Celerity and brought here. So thanks, Peter. Here and online to Nick for starting this community, I think 6 months ago. It's already got like 1,500 and growing people on it. So tell your friends if they're into this space to sign up for Bits in Bio. I'm sure we're going to do another one of these probably